the pleasure of hearing Leif Ellison speak and give the Salmon Lecture. The Salmon family gave money for this lecture in honor of Mrs. Salmon Frank, who was a patient actually of Dr. Matalonis's when she was treated here. And because of the importance of BRCA1 to this family, they endowed this lecture going forward. And this year, it is really our pleasure to have Dr. Ellison migrate all the way across town to come and give us this lecture, which can be more difficult than coming from out of town, as Leif well knows. So if you don't know Dr. Ellison, he is professor of medicine at Harvard Medical School and program director for breast medical oncology at the Mass General Cancer Center. And he is also co-leader of the breast cancer program for the Dana-Farber Harvard Cancer Center. Leif and I agreed that we would not tell you all of his training, but I promise he is truly Harvard trained once he left Stanford for many different categories. And now he has in his lab been working on breast cancer and on BRCA1 and 2, which he'll tell us about. And he's also done a lot of work on resistance to novel therapies. I don't know if that's part of this. So Leif, we're delighted to have you and to have you give the Salmon Lecture this year.
Thank you very much, Judy. It's a real pleasure to be here and an honor to be giving the Salmon Lecture. At the last minute, I changed my title, which initially was Early Pathogenesis of BRCA-Associated Breast Cancer. And the reason was that I thought it was a little bit too much. Saying pathogenesis, kind of implied that maybe we had a clear vision and overriding view of what's happening in early pathogenesis. And as will become abundantly clear this afternoon, that's not the case. And so I changed the title to Early Phenotypes of BRCA2/1 associated breast cancer. The idea that we're starting to understand pieces of the puzzle, but it's a little bit like the blind man and the elephant. I think we're still each seeing our individual pieces, and we need a little bit more work to fully see the whole. But I think we're going to get there, and hopefully some of the work that I'll tell you about will give you a clue about how we think we're going to do that. So the reason that we're here and the reason why BRCA1 and 2 are so important is because of the high penetrance of breast cancer as well as other cancers, including ovarian cancer, pancreas cancer, particularly for BRCA2, prostate cancer in men, and others. And I won't go into much detail about the clinical management of the challenge of folks who harbor BRCA1 and 2 mutations, but I will focus on one observation, epidemiologic relevant to breast cancer, that sometimes gets overlooked, and that's illustrated in these predisposition curves. And if a woman, 30, 35 years old, gets found to possess a BRCA1 or 2 mutation, yes, you can tell that patient that she has a 60%, 80% chance of developing breast cancer over the next 40 years. But in fact, that information is absolutely useless for that woman because it doesn't tell her anything about what's going to happen in the near term. And we know that over 40 years, basically everything is going to change. What would be more important for those women is, what is my chance of getting breast cancer in 1 year, in 2 years, in 5 years? And that's what these curves illustrate, is that the annualized incidence or predisposition of breast cancer in BRCA1 and 2 carriers is relatively stable over time. It's relatively linear. On average, about 2% per year between 30 and 70 for BRCA1 carriers, and about 1.5% per year for BRCA2 carriers. And that stability over time means that, for example, if a woman wants to make decisions about preventive therapy, there's no good time, right? You can't tell her that, your risk is mostly much later in life, and so don't worry. Her risk of getting cancer in the near term on an annualized basis is equal to that in many years from now. And so the absence of any near-term risk markers is one of the challenges that we had. And secondly, we don't have any very effective way to prevent these cancers. And so understanding what's happening before the cancers develop, we hope will be the key to bringing those solutions to our patients. So again, to underscore what are the challenges? Biologically, it's been even debated whether there is something called a precancerous state if one inherits a heterozygous mutation in BRCA1 and 2. In other words, is the loss of the other allele of BRCA1, 2 in the classic tumor suppressor paradigm just a random event, and it's just more likely to occur if you only have one good copy to spare rather than two good copies? Or is there actually a precancerous state that the heterozygous loss of BRCA predisposes to DNA damage and other events that might ultimately culminate in the loss of heterozygosity, which is thought to be important for invasive tumor development? I hope that I can convince you during the course of the lecture that it's really the latter case, that we believe there is a phenotype in the heterozygous state, which I'll refer to by its somewhat technical term of a haploinsufficient phenotype for BRCA1 and BRCA2. And so the underlying premise is that understanding that haploinsufficient phenotype might give us clues to prevention. Furthermore, understanding that even if we don't know all the details, might lead us to some of these near-term biomarkers of risk. What are the things that are happening within this haploinsufficient phenotype that could be quantitative in terms of a clinical biomarker that could give a woman an idea about, well, you have the biomarker, meaning that you're a lot more likely to develop your cancer in the next 5 years as opposed to 30 years from now. We have no such biomarkers now. So we hope that these kinds of studies are going to unlock the key to understanding of this kind. So before I get into many of the details about what's happening with BRCA and what we're thinking, being the blind man with the elephant, what I thought I would first talk about is what we hope to do with this information. And if you think, for example, about cancer prevention, and Dr. Garber and others are real experts in this area, and we know that to launch a cancer prevention trial, incredibly expensive and incredibly long-term, right? This is a once-in-a-career thing, and if you bet the wrong thing in your career, right, that's not a good thing. And so there's a big disincentive to do large-scale prevention studies. So my firm belief is that in order to do really effective prevention studies and do them right, we have to know more in terms of the specific hypotheses about what will be effective prevention. And one of the ways that we're going to generate and test those hypotheses is through what are called window of opportunity clinical trials. And that is the case where you give a patient a short-term exposure to a drug with a specific hypothesis about the effect of that drug on the tissue at hand, and then in many cases this is done in a patient who is awaiting some kind of surgery, and then you can assess the biological effect at the time of the surgery. And so this is particularly amenable for breast cancer, where there's a tumor in place, and you can look at the effect. And in this case, this was a study done by Laura Spring, published a couple years ago, where she used a PARP inhibitor known to be effective in advanced BRCA1/2 associated breast cancer as preoperative treatment for BRCA-associated early cancers confined to the breast. And what Laura was able to show was that just with the single agent PARP inhibitor, with short-term exposure, just 4 cycles, up to 40% of the patients had complete disappearance of the tumor. So again, this is not an FDA-approved level study. This is a hypothesis testing study that suggests that there's really quite remarkable activity of PARP inhibitors. We know that pathologic complete response, for example, is associated with long-term disease-free survival. So the idea that this is giving us a window into the potential effectiveness of PARP inhibitors in this setting. This was in a setting of a patient with a tumor in place who was a BRCA carrier patient, but you could equally imagine any BRCA carrier who hasn't developed cancer with his existing haploinsufficient phenotype that I'm going to tell you about, with exposure to an agent where we can test the effect of that agent, having a hypothesis about what it's thought to do, and if indeed we could see the intended effect, that would lead us on toward the path to potentially having the wherewithal and the courage and the funding perhaps to do these long-term prevention studies that we'll need to do. So I think this is really something to keep in mind as we think about what is the goal of all this important understanding we have about early pathogenesis of BRCA1 and 2. As we were thinking about how do we carry out these studies in our own institutions, one of the things that became apparent was that one of the major barriers to doing window of opportunity trials was actually the pre-treatment biopsy. So for example, if a patient's diagnosed with diagnosed with a cancer and comes to clinic, or a patient is found to be a BRCA carrier and comes to clinic, and you want to enroll a patient in a trial, say they're waiting a surgery for their cancer, a surgery for their preventive surgery, if you say to that patient, we have these great studies, but it's going to take a couple weeks. We'd like to schedule an extra biopsy, and we'll let you know, at some point when that biopsy is, and then you'll have to come in again and find parking and have this extra biopsy, and then if all that works out, then maybe you can think about entering the study. Huge disincentive to do anything, but yet this pre-treatment tissue is absolutely essential to interpreting the results of all these studies. And so a couple years ago, it occurred to me how valuable it could be if systematically we asked patients to participate in collecting collecting biopsies at the time of their diagnostic biopsy. It turns out that my office at Mass General looks right out on the radiology suite where all the biopsies were done. Our chair of breast radiology at that time, Connie Lehman, I made the pitch to her about how if we could systematically ask patients if they were interested in participating at the time of the biopsy. They don't even have an extra stick. It's an extra pass of the needle in through that same place where the first needle went in. Takes about 2 minutes extra time, and then we have the research biopsies on all the patients who might enroll in prevention studies, in treatment studies, and then all that tissue to analyze. And at first there was some skepticism. These are not cancer center patients, right? They may not even be Mass General patients on a regular basis. They've come in because they had abnormal mammogram and they were told they needed a biopsy. But in fact, our patients are very generous, and 2/3 of the patients that we asked gave informed consent for those extra passes, for those research biopsy collections. And so we had over 2/3 uptake of patients in consenting to that. This is a study we published a few years ago with the initial experience with a 0 complication, extra complication rate for patients who enrolled. To date, we've enrolled nearly 1,000 patients and more than 1,000 biopsies, about 1/3 of which are malignant. We have pre-malignant, we have risk lesions, we have all the benign lesions. And this is turning out to be a tremendous research resource for so much of what we do. And we think in particular that the kind of window studies that will be ultimately the output of the hypotheses and data that I'm going to just show you might be useful for. We also went on just to show, about how valuable these biopsies are. Literally, almost every month, new technologies are emerged that we can apply to these kind of biopsies. This was some examples of the paper on the left. You can see how we were able to look at HER2 heterogeneity, which is now known to be important for response to HER2-directed therapy of HER2-positive breast cancer. On the right, the single-cell RNA sequencing. We get thousands and thousands of cells, even from these very small biopsies for single-cell analysis. And then I'm not showing you any of the beautiful images of some of the spatial analysis that we're doing with Sandra Santagata and Peter Sorger. That's a whole different talk. But literally, it's no exaggeration to say that every day these biopsies become more valuable and we can learn more from them. So having them is a great resource. So all that's by way of introduction about the data I'm going to show you and thinking about how we can apply that to understand early pathogenesis and ultimately reach our clinical goals. So to do this, we were fortunate that there was a call by a foundation called the Gray Foundation that was very interested in BRCA1/2 associated cancers. And I was fortunate to be able to pull together a group of the most top researchers in diverse fields: computational biology, clinical management of BRCA, DNA repair, and genomics, to help in this effort. And so all the work that I'm going to be telling you about is really part of this team collaborative effort, which has been initially funded by the Gray Foundation, as well as funding from Department of Defense and other organizations. But really a tremendous team, and I put their faces up front because so much of it is their work. So our studies started based on the fact that we're very fortunate that Mass General is an international referral center for women seeking preventive mastectomy in the setting of BRCA1/2 and other high-risk mutations. And so, again, through the generosity of our patients who signed informed consent for us to analyze the tissue that would otherwise be discarded at this time of surgery, we were able to initially start some fairly primitive analyses where we could take the tissues, we could dissociate the cells, and then use flow cytometry sort them into different subpopulations and then analyze those populations through various omics analysis and functional assays. And that's what we started, more than 5 years ago now. And one of the reasons why being able to sort them in this way was important was because for those of you who know the BRCA field, you know that BRCA1 and 2, although they function in a common DNA repair pathway associated with homologous recombination-mediated repair, the phenotypes of the cancers are in some ways quite distinct, where about 80% of women with BRCA1 mutations developing triple-negative breast cancer, hormone receptor negative and HER2 negative, while those with BRCA2 cancers predominantly hormone receptor positive cancers. Why that is, we don't have a complete understanding, but it probably relates to somewhat different target cell of transformation in the breast. So studying these individual cell populations and how they may differ in the context of mutations and non-mutation is quite important. The first study that we did was literally just counting the proportion of cells. On the left, you can see flow cytometry dot plots where we can use markers to segregate epithelial and stromal cells into different populations. And among the key populations in the breast are, because it's a bilayer, are the luminal cells. And luminal cells basically segregate into two types. You can see them here very clearly in this BRCA2 carrier on the lower left. Mature luminal cells, also called now hormone sensing cells, luminal progenitor cells, now sometimes called luminal secretory cells. But these two luminal populations, and then in the bottom right here, basal epithelial cells, and on the bottom left is the stroma. And so simply by enumerating, it appeared that with age, in BRCA2 carriers, there seemed to be a predominance of the luminal over the basal lineage, where the proportion of luminal progenitor cells was increasing with age, but not so in those who were wild-type carriers, and that was at the expense of basal cells. And therefore, for technical reasons, you can actually look at the luminal-basal ratio, which is actually more robust, and showed that tended to increase over time in BRCA carriers, but not in non-carriers. This was of interest because, in particular, it was thought that the cell of origin, the key target cell for transformation and sequential events in the BRCA population, are these luminal progenitor cells, right? These are the cells during lactation. They undergo dramatic proliferation and differentiation to execute the lactation program, so they have this great proliferative potential, and they're thought to be under extra replication stress, which I'll talk quite a bit about in some of the future slides. So interesting that we saw an emergence of a greater population of some of the luminal progenitor cells in this small series in the carriers. But what was most definitive about showing that there really was something going on in the carriers that wasn't going on in the non-carriers was when we started looking at the individual cell level. And again, our fundamentals of cancer biology, we know cancer is a clonal disease. We think that pre-cancer is not clonal. And one of the biggest challenges in the setting of pre-cancer is that every cell is going to have something different. And so, that makes it difficult, because if we just grind up the whole tissue and then look, everything gets averaged out, we don't see it. But then secondly, even when we see things at the level of individual cells, it can be hard to find meaning. Nonetheless, what we did observe was that in the BRCA carriers, what we're doing here is now taking each individual cell and doing what's called low-pass whole genome sequencing. So for example, if you just look at the top row, the genome is on the x-axis here, each chromosome here, and the middle line, the gray line, means that it's diploid, right, normal copy number, and then you can see these color-coded areas where, based on the sequencing, even at low-pass sequencing, you can see gains and loss of chromosomal material. So the example here on the left is chromosome 1. The cell from one patient at the top, patient number 128, had the complete loss of the long arm of chromosome 1, and another cell from that same patient had a gain of the entire long arm of chromosome 1. And then another cell, this cell 129, also had loss of the long arm of chromosome 17. So you can see the blue is losses, the red is gains, and these seem to be frequent in these BRCA carriers. And then, interestingly, in some cases, we could see what appeared to be clones. You can see the similarity in this blue loss, which seemed non-random, suggesting that these might have been clones that were dividing, and an emerging tiny subclone evident in that tissue. And if you summarize the data that we published in the paper on this a number of years ago now, so we've really zoomed out, and this whole chart shows 250 cells from both BRCA2 and wild type. The BRCA2 carriers are on top. We've boxed the losses and gains among those cells. Here on the right, you can see the individual patients. And what we can see is that the cells of the BRCA carriers, over 20% had losses and gains, over 20% of the individual cells, losses and gains of chromosome pieces. And secondly, when we looked at non-carriers, a small series validated in larger numbers, we see very few of those. Secondly, those abnormalities seem to be restricted mostly to those luminal progenitor cells, which I mentioned earlier were the putative cell of origin for the BRCA-associated transformation. So this was a quantifiable and, you got the sequence, relatively concrete phenotype. And I put it here in the title so I didn't forget to tell you. The question was, is this classical tumor suppressor? We have LOH, maybe failure of homologous recombination, or not. And it turns out that in the cases where we could interrogate the individual cells, there was not loss of heterozygosity, being that indeed, whatever was going on, this seemed to be, by definition, a haploinsufficient phenotype in the cells of these carriers. I should also say that we're very fortunate to work with very talented and expert breast pathologists, and all the tissues that we did in the studies and subsequent studies were deemed to be histologically normal in these carriers. And other data more recently has suggested that even in non-carriers, risk lesions, atypical hyperplasias, and other types of lesions might have some of these same abnormalities. But in BRCA, it's more widespread, and it occurs in the normal epithelial cells, and so appears to be a distinctive phenotype. But the next thing that arose was, you have all these dots and spots and tiny losses and gains, and, is it really real? So over time, we've now adapted our methodologies to do this in a more robust way to try to get more information from this context. And I just put that here to give you an idea about some of the controls that we're doing to really look in detail about this damage and what's going on. And so here's a case where I plotted it in a different way. We're now seeing blue and red for losses and gains, but here again on the right, all these individual cells that we're doing this genome sequencing. Here we're sequencing a little bit deeper. And in this case, what we did, and this is the important part shown in the red arrows, we spiked in some non-cancerous but immortal cell lines called MCF10A that has known losses and gains, and then we sequenced everything in batch to say, okay, do we find everything that we expect in MCF10A reproducibly and nothing more and nothing less when we sequence together with all these patient cells? And fortunately, the answer was yes. So MCF10A, you can see these gains of chromosome 1, for example, it's always there. And then these tiny losses, chromosome 6, chromosome 9, it's always there. So we feel really quite confident about our ability to call and identify these small losses. So, and these are the patients, so when we see these small loss here, bigger loss here, small loss here, big loss here of the whole chromosome, we're really quite confident in that damage being real. Furthermore, this is a little bit early days for this analysis, but we're now able to sequence more deeply in these cases, and actually we believe we're going to be able to call mutation signatures. These are patterns of SNPs and indels, so nucleotide polymorphisms and insertions, deletions, that collectively might make up a BRCA-associated phenotype. Normally, with this very low-pass DNA sequencing, you can't call these because you don't have enough sequences for the statistical calls, but cumulatively, with the level of sequencing we're doing now, we think we may be able to call these signatures. But what makes us think these signatures are going to be here? This is chunks of chromosomes missing. This is a different process probably than the signatures. What makes us think the signatures are going to be there? Well, that comes from one of our partners in this collaboration, Ashok Venkatraman, who is literally the world expert on BRCA2. And we've worked with Ashok during the project to create ex vivo models using CRISPR to either knock out BRCA, and in this case BRCA2, or knock in various BRCA mutants. And I'm going to show you some data from his group throughout the talk. But this piece of data speaks to this issue of haploinsufficiency, like the mutant tissues, and mutational signatures. And what Ashok did was do a haploid knockout of BRCA, then compared to a homozygous BRCA CRISPR knockout, and then look for this BRCA-associated mutational signature. But there was a twist because, we don't believe that in vitro is exactly the same as in vivo. We believe that in vivo there are certain types of stresses that converge to create this tumor-stressed environment in which tumors arise. And Ashok believes, through many publications he's had, that part of that has to do with metabolic stress that leads actually to degradation of the remaining BRCA in the absence of loss of heterozygosity. So all that to say, the experiment was you have this heterozygous knockout, you treat with a metabolic stress, and then you see whether you can evoke this signature which you would normally associate with homozygous BRCA loss. So here's the control with homozygous BRCA loss. You get this, and it's this blue, a teal color. Let's call it teal. Here you get the teal signature here. But indeed, he can also see that in the haploid BRCA loss with the metabolic stress. And here you can see both day 17, day 35 of these ex vivo cultured haploid BRCA cells. Indeed, you can elicit that. And so we believe that that's a clue that maybe with this type of metabolic stress that we postulate is happening in vivo, we may be able to see that. Again, we don't know, it's early days, but we are very anxious to find out. And why is this important? Because it will really give us insights into some of the mechanisms associated with haploinsufficiency. So the other thing about the new work that we're doing that unfortunately I'm not going to be able to tell you the conclusion of is that now in those same cells we have this DNA, and getting deeper and deeper with the DNA sequencing, we can also do RNA sequencing, RNA-seq, comprehensive RNA-seq of the same individual cells. And to appreciate why that's important, you almost have to get to the end of the talk where I tell you that the real knowledge is going to come from getting much bigger numbers so that we can eliminate the heterogeneity between patients that is a huge challenge in all these studies of early pathogenesis. In other words, all of the things that converge to regulate cancer predisposition, the age of onset of the cancers, are reflected in the tissues. So if you just pick random patients, even if it's random BRCA patients, there's just too much heterogeneity to understand what's going on. In this case, however, the idea and the experiment is we are comparing within the patient to compare the damaged cells to the non-damaged cells within the patients to identify signatures associated with the damage. Now, it could be that these signatures are going to be meaningful in terms of mechanism, or it could be that the signatures may not be meaningful in terms of mechanism, but might hallmark damaged cells. Either way, it's conceivable that this, what we call G&T, simultaneous genome transcriptome analysis of the same cells, may give us potentially biomarkers of the damaged cells that could be applied clinically and to large data sets and to patients on a routine basis to understand the prevalence of these damaged cell populations, how they change over time, and whether they can be associated with future or near-term cancer risk as we postulate. And we think that that will be very valuable. So all this just to say that, we've shown here in our test set, right, that if you do the whole genome, whole transcriptome, you can easily identify all these key signatures of MTF10A that differs from the primary patient cells. And now we're in the status of ramping up to now look at damaged, undamaged cells from the same individual patients, and we're very excited about what we might learn from that. So, but what's causing this, right? I showed you this damage and chunks of chromosomes missing. What on earth is causing that? We believe that it's related to a replication stress phenotype. This is some early data from the original paper, and Miraban Caravaz really gets credit for all this work. And she was able to show that under replication stress, which had been postulated to be part of what BRCA protects against, she could elicit DNA damage in primary cells that were cultured. And this is a common assay where you run a current through individual cells, and therefore the broken DNA, you see it as a tail with the common. So you saw more tails under replication stress with BRCA mutants, this is BRCA cells right out of the patient, than you did in wild-type cells. And then if we looked at bulk RNA sequencing from these patients, we could see a signature that had been identified from lab studies where you knocked out the replication stress checkpoint. If you knocked it out, a certain signature was elicited under replication stress called the RSID, replication stress deficiency signature. And when you knocked at those things, you had this. We saw it in bulk RNA-seq from these same patients. Interestingly, if you do fewer genes and shallower sequencing with single-cell RNA-seq, it's a bit harder to see, but in bulk RNA-seq, you can see this replication stress deficiency. So we see stress-induced damage, we see a replication stress deficiency. Maybe that does mean something. Mirabon then went on to show that in these same primary cells, you could see a defect in the replication stress checkpoint, kinase check 1, and a failure to activate that checkpoint under replication stress. Again, these are primary cultured cells where we're using hydroxyurea as the replication stress. So all that, the idea that there is something different, it may relate to replication stress. Well, let's go into the details about what this replication stress is really about. So I'm not a DNA repair expert, but I will tell you that in the field, BRCA has a number of functions, BRCA1 and 2. The most prominent that you mostly have heard about is homologous recombination-mediated repair, but also, as I said, this effect of managing the replication fork, which is under many different stresses, and its integrity is absolutely essential. It's still a bit controversial whether, from the point of view of BRCA function, you can mechanistically separate the role in homologous recombination from the role of replication stress management. So, however, Ashok has done some work in this area using different BRCA mutants that are defective in somewhat different ways. And in these assays, he's using these same CRISPR knock-ins. This particular experiment is done with hemizygous cells, so it's just one allele. But he's using something called a DNA fiber assay, where you can interrogate replication by labeling the DNA strands, and then you can physically see them. And if there's a defect in management of the replication fork, the replication tract gets shorter, right? And so that's all you really need to know for the minute. I'll show you a little bit more in a second. But replication tract length is the readout. And if you have wild-type BRCA2 here, from left to right, you get a longer replication tract. The black line means a longer replication tract, so that's intact replication fork. And what Ashok was able to do was test a variety of mutants, all of whom were defective in replication tract maintenance, right? Meaning that replication stress was having a negative effect. And what he was then able to show, and the data's not here, that some of those mutants had fully lost homologous recombination, but some of them had not. And that suggests that indeed, in some ways, the function in replication stress and fork protection might be discernible or distinct from the effect in HR. And why is that important? It's important because much of our data points to the idea that it's really this part having to do with replication stress and fork integrity that could be the haploinsufficient phenotype, and I'm going to show you data specifically speaking to that, whereas HR, homologous recombination, is probably the phenotype that really requires LOH, complete loss of function, for BRCA to manifest. So this idea of replication stress management as part of the haploinsufficient phenotype. So let's look at what some of the data is for that. One of our other great collaborators, Lee Zhou, formerly at Mass General, now chair of biochemistry at Duke. And Lee was also an expert in doing these fiber assays and was started to ask the question, well, what is actually going on in the absence of BRCA that leads to this replication tract shortening, right? This failure at the fork. And this could be a lot of things. But what's now emerging, and this is, Lee has shown this here, but other people have shown it, part of it has to do with the repair of single-strand gaps that occur during replication. And so this is, again, this fiber assay. You label one strand as an internal control, then you label the second strand, and then when shortening takes place, that's an abnormal process. You can quantitate it here on the right. And in this case, what they're doing is looking at shortening that occurs when you treat with S1 nuclease, which specifically digests the single strands that normally get filled in during replication. And so what you see is these are ovarian cancer cells that have lost BRCA1. If you treat with S1, you get this dramatic shortening, meaning that there are these unfilled single-strand gaps. If you reconstitute BRCA1, that there are no gaps, right? S1 has no effect, meaning no unfilled single-strand gaps. So BRCA1, failure to fill gaps. This is in the homozygous state. Now let's look at the heterozygous state. And again, here we have our fiber assay. And so, again, part of our hypothesis has to do with this issue of replication stress. And what Lee is showing here is that in the heterozygous state, with one intact and expressed allele, you're still seeing a failure to fill in or repair these single-strand gaps. And you can see it here where you have this substantial difference in shortening between the heterozygous cells, particularly in the presence of replication stress, right? As I told you, if you compare lanes 1 and 3, replication stress itself leads to the shortening, and it gets exacerbated with S1, suggesting that part of that problem is failure to repair these single-strand gaps, and that it occurs in the heterozygous state under these conditions. So quite interesting if we're thinking about whether this could be part of the haploinsufficient phenotype. But you might ask, well, what about the chunks of chromosomes missing? Well, of course, the key is that if you fail to fill in these single-strand gaps, that creates a stalled replication fork, triggers checkpoints, and if those are not managed properly, it can indeed lead to single-strand and ultimately double-strand breaks, which if not prepared properly, could indeed give rise to the types of losses and gains that I showed you that we see in the patient's cells. This was articulated very beautifully in a recent review about the replication functions of BRCA1 and 2. So at least at a first pass, a plausible model. So much of what we're doing now is to try to flesh out this model to see how much of it we can see in the primary cells and see how our models are recapitulating what we think is going on in vivo. Finally, the question is, well, can we do anything about it? Is it possible that the haploinsufficient phenotype not only causes damage but also creates vulnerabilities? So this is an example suggesting that might be true. Again, work done by Lee, where he's shown that in the middle you can see the cells that are haploinsufficient for BRCA1, that have that failure of the replication stress, the failure of gap repair, indeed have an increased sensitivity to inhibitor of the replication stress checkpoint kinase and repair kinase ATR. And you can see it reflected in the shortening here of the tracts, and that it's associated with a failure of the checkpoint that we actually don't fully understand, but just an enticing clue that indeed a haploinsufficient phenotype could be associated with potentially therapeutically targetable vulnerabilities in order to perhaps either induce repair or elimination of damaged cells, potentially in the future in carriers of BRCA. So then we went on to characterize these other phenotypes, right? And here we're looking at transcriptional phenotypes. And this is work that we've been doing with Gad Getz, Nick Hardvala and his group, where we're doing single-cell RNA-seq on cohorts of patients. You can see here left a UMAP looking at all the cell types that we see in the normal breast that we can enumerate in BRCA and wild-type non-carrier patients. One of the interesting things came out had to do with the number of T cells and immune cells in these cases. And that sparked a collaboration with Sean Demiri, who is very interested in immunoprevention. And so Sean said, well, in your single-cell RNA-seq, you're seeing something that may be different in the T cells. We should start looking at those. And what he showed subsequently was that in the lobules themselves, you see an increase in T cells in both BRCA1 and 2 carriers, and particularly CD8 cells in BRCA2 carriers. Sean has gone on to make a number of inbred models where he can test the idea that there is an altered immune response to the cancers. This is one of his models where he's using a heterozygous BRCA knockout mouse with K14 in a heterozygous state, as I said. Then he used these stress conditions to induce tumorigenesis with high-dose estrogen and low-dose radiation to invoke these spontaneous mammary tumors. And what he sees is that in the setting of BRCA, you have really a fundamentally different immune environment. He's gone on to characterize in more detail the actual microenvironment that's going on, the state of the CD3 cells that are involved in these tumors, and then, backing up, in the pathogenesis of these mouse tumors to try to understand the role that the immune system might be playing in this context. Is the immune system inflammatory in the setting of DNA damage and a cancer predisposer, or is the immune system protective and an immunosurveillance in that way? And I think Sean's models are going to give us important insights to that. But I think this is one piece of data that suggests that this is a model where the phenotype is present. Then, if we're focusing back on our epithelial cells, which was really, our cells of interest here, even with small numbers of patients, we start seeing signatures that seem to be relatively distinct in the BRCA carriers. So on the left, this is just a UMAP of, now we're zooming in on luminal progenitor cells, where you can see clusters of cells associated with the BRCA carriers that harbor certain profiles of transcription. And particularly, those were profiles enriched for a number of stress-induced and epithelial transcription factors and of a particular growth factor, c-KIT, otherwise known as KIT, which was shown to be higher in the BRCA mutants than in the non-mutants. And we call this the KIT signature because it seems to be a cohesive signature that goes together and is enriched in the luminal progenitor cells of the BRCA carriers. Anytime we see something like this, because it's a relatively small number of patients, we like to test it in other ways and go back to our models. And so here, for example, we can look at a much larger database we have of bulk RNA-seq from these patients and show that we're seeing essentially the same sorts of things, right? That we're seeing more of the c-KIT positives, that's shown in blue, and that we can actually see it, the pattern emerging in bulk RNA-seq through distinct and other cases that were processed in a completely different way. The same pattern emerges. Now, is there anything here that's of interest or potentially actionable? Well, c-KIT itself certainly has gotten attention in the setting of BRCA, and this is work from many years ago that suggested that indeed c-KIT and its signaling pathway was enriched in human epithelial cells, particularly in BRCA mutants, that if you looked at c-KIT-associated phenotypes, if you remove these cells and culture them, that both in patient cells and in mouse models of BRCA, that you see an enrichment for c-KIT signaling, and that somehow the c-KIT axis might be important in some of these early pathogenic steps. And the idea is that the deregulated signaling might have an impact on some of the replication stress and other phenotypes that we think are important for DNA damage. In particular, one of the key phenotypes that emerge from the cells, both in the models and in the primary cells, related to the c-KIT phenotype was a DNA damage UV response, which may not be so surprising, but also a strong ROS signature. So these are various signatures where we're comparing signatures in various BRCA mutant models, again comparing to the primary patient cells. This is particularly in BRCA1, where consistently you see enrichment of ROS as well as DNA damage response signatures in the setting of the mutant heterozygous context. And why could this possibly be important? Well, again, this is back to work from Ashok, where his work has suggested that indeed ROS is a key feature associated with BRCA2 deficiency. And Ashok, in a recent paper a few years ago in Embo Molecular Medicine, has shown that loss of BRCA2 induces mitochondrial dysfunction associated with ROS features that, although the precise steps related to BRCA and the mitochondria are not entirely clear, indeed provokes increased oxidative stress. And so you can imagine where we're talking about oxidative stress in this tissue compartment that's already under other stresses, such as hormonal stress that we know is so key to breast cancer pathogenesis, that this deregulated response and handling of oxidative stress could be part of the process that's ongoing and may ultimately be a cause and maybe even part of an effect of the DNA damage we see. And this sort of concept is validated again in our models and again in the setting of BRCA1 as well as BRCA2 when we use back to our CRISPR models now for heterozygous BRCA1, and we look over time in the same way that Ashok was looking at those signatures with his metabolic stress. Here that was BRCA2. Here in BRCA1, what we see under certain conditions is the emergence of an ROS signature in these models, specifically of BRCA1, that does not emerge ever in the wild-type cells. Right? This is a very characterized. This MCF10A, which is a very well-characterized system for analyzing these kind of mutation profiles. And MCF10A particularly good because they're not particularly genomically unstable as non-transformed cells and suitable to these kind of mutational process analyses. And so, we believe that this says that indeed the ROS signature, ROS handling may be part of the phenotype that we've uncovered here. And so this is from Po-Han Lin, who is a very talented Taiwanese physician scientist in the group, was sitting down one day doodling, and I think this is a literal copy of the doodle. So it's not meant to be taking anyone's Bible, but I think that it gives you some kind of clues about how we're thinking about how some of these pieces of the puzzle might fit together, related to defective OXPHOS and ROS handling. Hold on, I can't see my thing. Oh, well. Oh, sorry. My screen went blank. Let's see what I have to do here. That's all right. Defective OXPHOS handling and ROS, and that may be exacerbated by the enhanced signaling through factors like KIT. That defective OXPHOS may lead to replication stress that we know is likely to be part of the DNA damage that we're seeing and that I've shown you, and that certainly cell proliferation may be part of the reason why the luminal progenitor, the proliferative compartment, is so predisposed to that, and that's going to give rise to a series of events, probably multiple genomic insults, that will ultimately culminate in loss of heterozygosity of BRCA1 and 2. And then once BRCA1, 2 LOH occurs, then you've really accelerated a genomic mutational process having to do with loss of homologous recombination that probably drives the final steps in the pathogenesis of invasive cancers, right? So again, when I talked about the blind man and the elephant, you can see how it relates to this. But I think it's a construct to think about how we want to work. But again, I think in terms of true understanding and truth, the patients are going to be in some ways the most informative model, but we need more. And so we're very fortunate to have participated and joined another great collaboration to increase the scale and throughput of all the types of assays and experiments that I've showed you. And this is culminating in the generation of the BRCA Precancer Atlas, the idea being to systematically reveal early mechanisms and actionable pathways. And this is a great collaboration between our group, Joan Brugge's group at Harvard Medical School, Kate Nathanson's group at Penn, the group here with Judy Garber and DFCI, and the tissues that are being collected that are pooled. And really, it is becoming a really multi-omic tour de force of single-cell RNA sequencing, DNA sequencing, attack sequencing, high-depth targeted sequencing, multiplex spatial analysis, all linked to the patient data, where we're going to have large enough numbers to segregate the patients into specific cohorts, which we think are going to be quite important to understand BRCA phenotypes for the reasons I told you, having to do with the issue of how age, hormonal exposure, parity, menopause all have major effects on what we see in the tissues. Dividing these individual cohorts and getting enough patients, BRCA2 and non-BRCA2, good controls to be able to do all this, we really think is going to be absolutely essential to a true understanding of what's happening along the lines of the sort of hypothesis generating and testing work that I've shown you. So this is going to be, a big effort over the next probably 5 years to generate data. But, we'd like to get to the clinical endpoints faster. So one of the parts that we're quite focused on is taking the knowledge and translating it rapidly into biomarkers that could be tested as these near-term risk preventers or at least phenotype correlates in BRCA carriers. And there you need very big numbers. And that means going back to our archives, not just enrolling patients prospectively, but in looking at every patient who's come through the door where we can get the IRB approval to look at the tissue, creating ththese tissue microarrays, right? You have spots of tissue where you can look at dozens and dozens of patients on an individual slide, and in that way to be able to interrogate hundreds and hundreds of patients through with the putative biomarkers that are coming up using all these advanced advanced technologies for spatial and single-cell imaging that we can now apply in this way, and then correlating these with the patient outcomes, with the future cancer risk, with the phenotypes, and with the demographics associated with that, to really get an understanding of what are likely to be informative markers that we could apply to tissues going forward. And we really do believe that this sort of concerted effort on laboratory-based systematic studies and a focus on clinical biomarkers is the fastest way to make progress in a meaningful way. So I'll summarize everything that I've told you. Identifying damage that precedes LOH in histologically normal tissues forms the basis for studies of risk biomarkers and prevention targets. Understanding the context-specific mutational processes and inciting factors paves the way for interventions to mitigate patients' risk. That coupling these functional analyses with hypothesis-based pharmacologic testing provides the promise of correcting or eliminating DNA damage, precancerous cells, potentially as mechanisms for prevention. And that phenotype-focused biomarker discoveries should enhance the efficiency and accelerate practical application for these patients. So thank you for your attention. I can't stop without acknowledging everyone. I acknowledge a lot of our collaborators. These are our many of our proximal collaborators, and I know there are many people missing, for which I apologize. But I'm always thankful to all of our collaborators, and particularly thankful to the patients who are always so generous in participating together with them in these studies to improve outcomes for them and their families. So thanks very much for your attention. I'm happy to take questions.